Hello, my name is Sneha Saha, and today I'm going to present the article Clear Proteomics reveals the molecular basis of the efficiency of human recombinant nerve growth factor treatment for neurotrophic keratopathy. Here, here, the corresponding author's name is Damiana Piergostino. She has affiliations from the Center for Advanced Studies and Technology and in the Department of Innovative Technologies in Medicine and Dentistry, both from the University of Gabriele di Annunzio from Chieti, Italy. She has worked in the following areas of biochemistry as shown below. This is a picture of Damiana Piergostino. Next, this article mainly presents neurotropic keratopathy, known as NK, as a rare degenerative corneal disease. It is also known as an orphan disease, and it is characterized by epithelial instability and decreased corneal sensitivity due to the damage of the corneal nerves. The treatment with human recombinant nerve growth factor (rhNGF) eye drops has shown an increase in the length, density, and number of nerve fibers branches during treatment. This treatment has also led to an increase in the DNA synthesis, protein synthesis, and fiber regeneration. And all in all, this treatment has proven to be a much better alternative than neurotization surgery. Thus, the main topic is treatment of NK with clear proteomics. The research question 1 is, what is the role of clear proteome in treating NK patients at baseline, which is week 0, often referred to as timeline 0, and in the follow-up period, which is week 4, and at the end, which is week 8 after topical rhNGF treatment. Next is, what are the molecular repercussions of pharmacological treatment and what are the new molecular markers of response of this treatment? Now the objectives for answering such research questions are to investigate the role of clear proteome in treating NK patients at baseline and in the follow-up period after topical rhNGF treatment, to find out the new molecular repercussions of pharmacological treatment with rhNGF, and to find out new molecular markers of response to the rhNGF treatment. Here, we are now going to talk about the various methodologies that were presented in the article. The first one is patient enrollment and clinical evaluation during treatment. In this observational case study, 15 patients, among them 7 males and 8 females with severe or moderate NK were enrolled. Among them, only 8 of them finished therapy. All the patients were treated with Senerclip, which is a type of rhNGF eye drop in the affected eye, 1 drop every 2 hours, that is 6 times per day for 8 weeks. Next, tear sample collection and extraction for proteomic study. Here, the tears from the subjects were collected on graduated skerma strips. Proteomics analyses were executed on the whole lacrimal fluid from the 8 subjects at baseline T0 and after week 4 and week 8 of treatment with rhNGF. Next, proteomic analysis and data processing. Tryptic peptides from each sample were analyzed in triplicate by using a proxy and easy chromatographic system. Next is bioinformatics and functional analyzing. Bioinformatics analyses are performed using PERSIS software, uploading the identified protein groups generated by MaxQuant, another software. Now, Western blotting was carried out after tear extraction and protein quantification by Bradford assay. Tear proteins were separated by 10% SDS phase, transferred to a nitrocellulose membrane, and then blocked for 1 hour at room temperature using blocking buffer. The optical density of Western blotted bands were assessed through the ImageJ software by calculating the percent mean of pixel intensity. Next is the flow of cytometry detection and subtyping of extracellular vesicles. The tears of the subjects were stained using a reagent mix. Then the samples were incubated for 45 minutes at room temperature in the dark. For all use parameters, height signals and the logarithmic modes were selected. Lastly, statistical analysis was carried out by using the Kruskal-Wallis test with Dunn's multiple comparison post-test between the three clinical groups was performed using a GraphPad Prism software. Then the rank correlation between variables are performed using MedCalc by using a Sperman non-parametric correlation test. Now, next is the new techniques and instrumentation that was discussed in this article. Proteomic analysis that was executed on the cold lacrimal fluid at week 0, timeline 0, at week 4, timeline 4, and at the end, which is week 8, timeline 8 of treatment with RHNGs. Then we come to the results. Here, the table shown shows the unique proteins that were identified in the three clinical conditions: timeline 0, timeline 4, and timeline 8. Here, the gene lane, and which is also known as neutrophil elastase, and ACTBL2, which is beta acetylene-like protein 2, these two were discovered at week 4, when the rest of the 6 unique proteins were discovered at week 8. Next, clear functional proteomics result after week 4 of treatment is shown. Here, the first one shows the inflammation of organ, while the second diagram shows the DNA synthesis. These diagrams uh, were created by downstream analysis comparing the proteomics dataset at week 0 and week 4 condition. Next, this shows the distribution of clinical data. Here, diagram A shows the fiber length, diagram B shows the nerve density, diagram C shows the ramification of number of nerve ramifications, and diagram D shows the diameter of nerve fibers. This data is visualized as a beam plot with a polygon shape. It represents a density trace computed using a log transformation of each variable. Next, we move on to the key findings in the article. The first one is functional in vivo data demonstrated a condition of severe and high in situ inflammation in patients with NK during the RHNGF treatment, and this inflammation could be associated with to a specific triggered inflammation mechanism in response to the treatment. Now, from the 15 subjects originally enrolled, only 8 of them completed the study. The rest dropped the treatment due to significant inflammatory side effects. Next, after 4 weeks of treatment, the protein expression in tears is driven by activation of BDNF, brain-derived neuropathic factor, and VCN gene by activated upstream. Now, the numerous effects of BDNF on the nervous system include axonal regeneration, plasticity, remyelination, and these could be correlated with the morphological improvements described in the patients. Next, at the end of the treatment, as discussed before, the 6 unique proteins were found at the week 8, and they were mainly related to neuroprotection, immunity response, and inflammation. Then, after 8 weeks of treatment, upstream analysis allowed to predict a strong activation of IL-6R due to inflammatory stimulus. Then, the number of neuronal EVs, referred to as extracellular vesicles, which is CD171 plus, increases during treatment were related to the increase of EVs expressing IL-6R, which is CD126 plus. Furthermore, the number of neuronal-derived EVs expressing IL-6R increases proportionally to leukocyte-derived CD45 plus EVs. Next, length density and number of ramifications of nerve fibers increased during the treatment, while the diameter of nerve fibers were seen to increase as significantly at the end of the RHNGF treatment. The epithelial restoration and stromal healing during treatment are related both to an increase of the inflammation pathways mediated by EVs and an increase of the nerve fiber density. Therefore, in conclusion, NK patients presented good healing in terms of the length, the density, and number of ramifications of nerve fibers after RHNGF treatment. For the first time, evidence on molecular pathways involved in the response to therapy of NK patients was found, and the main evidence suggested that combining neurodegeneration and inflammation mechanisms during the treatment in some cases determined a serious inflammatory reaction that triggered a neurodegeneration resolution. Therefore, this data will contribute to a better understanding of the biological pathways underlying the therapeutic effect of RHNGF in the treatment of NK that could be useful in the assessment of drugs in the future. Thus, uh, the summary of the article is NK is a rare degenerative corneal disease. Treatment is possible using RHNGF eye drops, so particularly using Senargrim, and this treatment has proven to be a better and valid alternative to neurotization surgery. And this RHNGF treatment has increases the DNA synthesis by activation of both BDNF signal and IL-6 receptor, along with stimulating protein synthesis and fiber regeneration. Therefore, in the future, we can say that this article is of case series nature, and superior study designs like randomized control trial (RCT), case control study, prospective study needs to be done to find out the effectiveness of RHNGF in treatment of NK. Study will be planned with an appropriate sample size by assuming all assumptions of randomized control trial and by using multicentric trial. With this slide, I end my presentation. Thank you all for your patience hearing. Thank you.